reported latency reversal agent in people with treated HIV-1. So what is HIV? HIV is a retrovirus, which is a type of virus that uses RNA as its genetic material. Retroviruses carry a specific enzyme called reverse transcriptase to convert its RNA into DNA after infecting a host cell. That DNA is essentially integrated into the host cell's own genome, basically hijacking the cell to make more viruses upon replication. ART stands for antiretroviral therapy. It's a daily combination of drugs that stops the HIV-1 virus from multiplying by often controlling HIV-1 replication to levels below limits of detection, which ideally allows the immune system to recover, keeping people healthy. However, it must be taken lifelong despite significant cardiovascular and metabolic toxicities and a pretty expensive cost. RIL-2 stands for recombinant interleukin 2, which is a synthetic lab-made version of the cytokine IL-2, which is basically a signaling protein made by white blood cells that stimulates immune cells, particularly T cells, to grow and fight infections or cancer. Aldesleukin is the kind of RIL-2 that they use in the study, which is an immunomodulator, which is currently being used for the treatment of several cancers. However, because of its potency, has been investigated for a number of autoimmune diseases like HIV. So basically, when HIV infects the immune system, it mostly affects immune cells called CD4 plus T cells. HIV spreads in the body as these T cells replicate, and most HIV therapies like ART are targeted to treat these actively replicating cells. However, sometimes these once active T cells go into quiescence, becoming latently infected in their G0 or resting state. Basically, these cells are like they're asleep. The virus becomes transcriptionally silent and undetectable by ART as they are no longer actively replicating. Similarly, the virus can infect CD4 plus T cells already in the G0 state and TRM cells, both of which are essentially in this relatively inactive state. Together, the infected cells form the latent reservoir, which is the collection of immune cells that are asleep and inactive that harbor this hidden HIV-1 proviral DNA. The latent reservoir is dangerous because it forms a long-term storage site that ART cannot eliminate, and so if ART suddenly fails or stops, the reservoir provides the exact blueprint for HIV to replicate again, leading to viral rebound and disease progression to potentially even AIDS. And so the researchers want to eliminate this viral reservoir using the shock and kill strategy, which is basically when they use a latency reversal agent to essentially force the dormant virus to shock it to begin replicating and expressing itself, which would then ideally activate the immune system to complete the kill, recognizing and destroying these newly visible infected cells. So the researchers conducted a trial testing the potency of the RIL-2 drug, aldesleukin, as a shock and kill agent. So Table 1 describes who was enrolled in the study and how they were treated, which is crucial for interpreting how strong and generalizable the results are. The study included 9 participants, all men, on well-controlled HIV on ART, with undetectable or very low viral loads at screening. They had high baseline CD4 plus T cell counts, meaning they were immunologically stable. They'd been on ART for a long time, a median of about 10 years, and so that means any viral reactivation seen later is unlikely to be due to poor ART adherence. Serious participants received 1 to 3 cycles of recombinant IL-2 depending on tolerance. However, we did see serious adverse effects occur in 3 participants, all of whom had received over 1 cycle of RIL-2. So the researchers actually only ended up being able to look at patients who had received 1 cycle of RIL-2 in the study. Off? Hello. RIL-2 in the study. So overall, Table 1 establishes that IL-2 induced viral reactivation occurred in people with long-term viral suppression and strong immune systems. However, it highlights certain toxicities that limited the study, which is an important constraint for using IL-2 clinically. Figure 1 looks at the virological shock caused by RIL-2, how much basically HIV-1 RNA was released into the blood. So panels A and B and panels C and D measure the same things. They're just 2 different kinds of assays. Panels A and B are a standard clinical assay, and panels C and D are an ultra-sensitive single copy assay, which is just a little bit more specific. And so panels A and C measure the concentration of HIV-1 RNA in logarithmic copies per millimeter, which basically just allows them to measure a large range of data. The x-axis represents the time points. These you will see consistent throughout the study. Baseline represents the beginning of treatment. Day 7 is the peak response, which was about 3 days after the first treatment cycle ended, and the end of the study was about 23 weeks later after the first treatment cycle ended. And so what they saw was that while participants started with suppressed viral loads, their levels rose to a mean of 301 copies per milliliter by day 7, which is really good. Panels B and D measured the magnitude of the shock, looking at fold change over baseline. The numbers on the y-axis mean that if you have a 10, there would be a 10-fold increase, a 100 would be a 100-fold increase. And so what they saw was that an average amount of HIV-1 RNA increased by 26-fold in the standard assay and 19-fold in the ultrasensitive assay by day 7, confirming that the shock was successful and that the virus is no longer just sitting quietly in the DNA of resting cells, but actually had begun to actively replicate and shed into the bloodstream. It is important to note that this increase was transient, meaning that the viral loads returned to near baseline levels by the end of the study. This suggests that while the drug could wake up the virus, the participants' ART was still effective at eventually suppressing that newly active virus once the treatment cycle ended. In Figure 2, the researchers looked at the same things they did in Figure 1, but this time in the latent viral reservoir, looking at whether a single cycle of RIL-2 administration had an effect on the concentration of HIV-1 proviral RNA, basically whether the shock from Figure 1 led to a kill. So they used an intact provirus DNA assay to categorize the proviral DNA and separate it by quality. Intact proviruses are the near full-length sequences of viral DNA that contain all the necessary genetic information to produce new infectious viruses. And these are what the researchers wanted to kill with RIL-2. Panel A, they saw a slight decrease in intact proviral DNA, but there's no indication that it was statistically significant. They did, however, in panel B, which measured magnitude of change, like the last figure, see a statistically significant 1.2-fold decrease in total proviral DNA. This seems ideal. However, if we look at panel C, which measures the ratio of intact to total proviral DNA, we actually do not see a significant change. So because this ratio didn't change, the researchers realized the reservoir did not actually shrink and concluded it had been killed after one cycle of RIL-2. The infected cells were simply diluted in the blood sample. The researchers called this the dilutional effect. As we will see in Figure 3, RIL-2 administration caused the amount of circulating CD4 plus T cells to significantly increase. And so this influx into the bloodstream actually made the proportion of infected cells appear lower, even though the total pool of them likely remained the same. So Figure 3 provides a comprehensive look at how RIL-2 administration physically changed the population and behavior of T cells in the study participants. It tracks these changes from baseline to day 7 and throughout the end of the study. Normal CD4 plus T cells are helper T cells, coordinating the immune response by activating other cells to kill. And CD8 plus T cells are cytotoxic and directly kill infected cells. So panels A through C look at T cell population expansion and proliferation. Panel A shows that the number of circulating CD4 plus T cells doubled and CD8 plus T cells increased by 1.6-fold by day 7. Panel B confirms this as there's a 13-fold increase in the cell division marker Ki67, which basically shows that the cells were aggressively multiplying. Panel C is important because it shows a transient induction of regulatory T cells. These specific T cells are actually cells that suppress the body's immune responses to prevent overactivation or autoimmunity, but they are highly sensitive to IL-2. So while intended to boost the immune system, the drug also actually induced suppressor cells, which could be a major drawback of the study because they might actually shield the HIV-infected cells from being killed by the rest of the activated immune system, which is why we potentially don't see a reduction in the latent reservoir. Panels H, J, and L look at how the drug rejuvenated these immune cells. Panels H and J show that CD4 cells were becoming highly alert and less fatigued as surface expression of SLAM-F6, a marker of high cell activity, increased, and CD101, a marker associated with immune exhaustion, meaning the cells were no longer able to fight, decreased by day 7. Panel L shows a decrease in CD57, which is a marker of cell aging on CD8 plus T cells. This means that these killer cells were shifted towards a more youthful and proliferative state, which is what we want in the context of fighting a virus. Panels E and F look at panel E looks at the anti-apoptosis, which is cell death, protein called BCL2, showing a decrease in the percentage of BCL2 in both cell types by day 7. and F shows a decrease in PD-1, which is another exhaustion marker. However, this remains decreased even up to the end of the study, which suggests potential long-term effects of RIL-2 administration in rejuvenating these immune cells. So taken together, figure 3 shows that RIL-2 successfully recruited a massive army of T cells to fight HIV, as we saw in panels A and B, and upgraded their status to be more active and youthful, as we see in panels H through L. However, it also reveals two major hurdles: that the army was fragile and short-lived, as there was a reduction in a protein that prevents cell death, and it included a higher number of suppressor cells, the Tregs, that might prevent a successful kill. Figure 4 looks at the results of single-cell RNA sequencing that allowed the researchers to look at the genetic rewiring that occurred within T cells following RIL-2 treatment. And so the plots in panels G and I show which genes were significantly upregulated or downregulated after IL-2 treatment. The y-axis represents statistical significance. Both cell types contain the downregulation of the inhibitory gene KIR2DL1. Its primary job is actually to prevent T cells and NK cells from attacking, and so its downregulation is essentially removing these brakes, making the immune system more aggressive and ready to target the HIV reservoir. They also saw the upregulation of the genes G0S2 and ALDH7A1, which are genes involved in cell cycling and metabolism. All this is that it shows that the cells have moved out of a resting state into an active phase of growth and energy production. Panels H and J look at changes in the transcriptional pathways. In both cell types, they saw a strong upregulation of the interferon alpha response and oxidative phosphorylation pathways. Because interferons trigger antiviral defenses and the oxidative phosphorylation pathways increase cell metabolic processes, we see that the T cells are actually being primed to recognize and respond to HIV-1 viruses and are preparing to meet high energy demands, because T cells need fuel as they're dividing rapidly to kill. In CD4 plus T cells, we see the downregulation of pathways related to killing ability. This transcriptional decrease in their ability to perform killing functions shows that they are being primed for latency reversal, which is the main goal for CD4 plus T cells, as they're being pushed into a state that favors HIV transcription, or the shock part. In CD8 plus T cells, we see the upregulation of cytolytic pathways, which are pathways to kill, cell surface adhesion, which allows these T cells to attach to the cells that they want to kill, and glycolysis, which allows them to have enough fast energy to perform the killing. And so we see that these CD8 plus T cells are being pushed toward their killing capacity to kill the latent reservoir. Figure 5 focuses specifically on natural killer cells, which are white blood cells in the immune system that can rapidly identify and destroy virus-infected cells. These are similar to CD8 plus T cells. So they saw the upregulation of CCR, which is a surface marker that recruits immune cells to sites of inflammation or infection. And so their increase means that the NK cells have a higher ability to migrate towards sites of inflammation, which basically means they're better able to enter tissues where the HIV-1 reservoir is. Panel B looks at different subsets of NK cells called CD56 bright cells and CD56 dim cells. There's an expansion of the bright cells and a decrease in the dim cells, which happens because the bright cells are very sensitive to IL-2. Normally, bright cells have low cytotoxicity, while dim cells are actually highly cytotoxic killers. However, IL-2 actually causes the CD56 bright cells to become potent killing cells, and actually they become even superior to dim cells after IL-2. And so we want this shift in the context of autoimmune disease treatment, because the bright cells help control inflammation while effectively regulating T cell proliferation and also killing activated infected T cells. Too many dim cells would be too cytotoxic and would cause harmful inflammation. So we actually end up seeing a successful transition of the immune system toward an environment with more robust immune and cytokine responses. Basically, it's better able to kill the latent reservoir, driven by this expansion of the CD56 bright NK cells. Panels F through G show how the CD56 bright cells were becoming more cytotoxic. We actually see essentially the exact same genes being upregulated and downregulated as we saw in figure 4, showing that the NK cells are becoming more active and aggressive in targeting the HIV reservoir. And we also see the same transcriptional upregulating of the pathways that enhance their capacity to kill, like the CD8 plus T cells in figure 4. So overall, we see a shift toward killing in CD56 bright cells that are usually less cytotoxic, but have been transformed to perform similar metabolic and effector function as the killer CD8 plus T cells. And so the researchers concluded that RIL-2 was one of the most potent LRAs ever tested in humans. supported by the massive viral reactivation that they saw on day 7, as the plasma HIV-1 RNA levels rose by an average of 26-fold. And they saw an immune power-up of the immune system. Basically, the treatment caused a robust increase in activity and number of natural killer cells and T cells. These cells were specifically reprogrammed at a genetic level to enhance their killing pathways. And so this is significant because one of the biggest hurdles in HIV cure research is that even if you shock the virus out of hiding, the immune system is often too exhausted to actually kill it. And so the fact that the findings prove that our IL-2 provided a robust immunologic boost is really good, as it essentially transformed these CD8 plus T cells and the natural killer cells into a more aggressive and metabolically capable killing force, specifically designed to target the reservoir cells that the drug had just shocked into visibility. However, in spite of all of this, we do see a limited impact on the reservoir. Despite the successful shocking and arming of the killers, a single cycle of treatment didn't significantly reduce the overall size of the viral reservoir, which leads to the limitations of the study. We see a low number of study participants, and so the researchers really weren't able to generalize the results to a wider range of people, and there were pretty significant side effects and toxicity to taking the drug. The viral reservoir was not actually reduced because of this dilutional effect, which led the researchers to want to know whether the current induction of the CD4 plus T cells, the T regulatory cells, impairs the capacity of the activated immune cells to kill the infected CD4 plus T cells because of their suppressor function. They also wanted to know which specific cells or tissues were releasing the HIV-1 RNA into the blood. They thought that it actually could be coming from tissue reservoirs rather than circulating cells, which, if this is true, makes it a lot harder for the CD8 plus T cells and the NK cells to target the virus. And so this all kind of culminates in the need to know where in the body the latent reservoir is maintained, which would be a key factor to understanding how best to facilitate HIV-1 cure strategies. And so the researchers suggested new constructs of our IL-2 that would avoid the T regulatory activation and allow the drug to activate killer cells without the immune suppression, as well as investigation into where anatomically the latent reservoir is maintained in the body, as well as multiple RIL-2 cycles at lower dosages to prevent harmful effects, or RIL-2 in combination with other therapies to better implement the kill part of their shock and kill method. I'd like to thank the Journal Club staff and Mr. Maxwell for helping me with this presentation, and all the teachers for giving extra credit. Any questions?
Students first, don't be bashful. If you got a question, ask.
How did the researchers know that RIL-2 is an ethical drug to test on humans if it's so potent?
Because it actually is being used to treat certain cancers. Aldesleukin is used to treat, I think, metastatic melanoma and renal something cancer. And so they actually, the concentration of the drug that they used in this study was less than the dosages that they were giving to patients with cancer. So there was already a precedent. Dr. DeCilia.
Okay, I thought this was a really cool paper. So they only did one dose though, right? Is that correct, of the...
I think they did 1 to 3 cycles of RIL-2.
What does a cycle mean?
I'm pretty sure it's like a dosage, and then like one cycle would be a
And then they made it
dose.
a little and then another dose?
Yeah.
Okay. I'm just wondering if, like, it seems like over time, after a little while, we saw a pretty robust response, but then it just went back down. So did they talk at all about, like, hitting the cells again at that high response to try to maintain, like, longer?
Yeah, I think what they were worried about was that if they continued to use too much of the drug, it would be too toxic to the body, which is why I think in the discussion parts or in future directions in the paper, they talked about using RIL-2 at lower dosages and then more frequently to induce a better reaction.
Was it an IV administration?
Yeah. Okay, thank you.